Good afternoon, everyone. I hope you have enjoyed your lunch. We'll now be moving into the last session for this symposium, which has been wonderful so far, and I hope the last session will be wonderful as well. So my name's Musalula Sinkala. Most of the people call me Sinkala. I am coming from the computational biology division here at the University of Cape Town. So every once in a while, wonderful people like Joanne come down to our lab and pick me up from there. I spend most of my time looking at a computer screen, and my friend is a blinking cursor that tells me to input some code. But so when Joanne came to pick me up, I didn't like it. But then after I came here, because I had to leave my friend, the blinking cursor on my computer screen. But after I came here, I realized that this is the place to be. And I've loved it. And I wouldn't be anywhere else. So the session that I'll be sharing is on unearthing the ecosystem, hidden ecosystem, and it will be focused on artificial intelligence / machine learning and informatics, and how we can apply this to mine for insights from microbiome data. And I'll start by, first of all, describing what artificial intelligence is. It is a field that involves training machines or systems to think like humans, whereas AI is a subfield of artificial intelligence. So artificial intelligence is a bigger field, AI is a smaller field that involves training machines on data to achieve particular tasks. I'll start by first highlighting how this transformative technology is going to revolutionize biomedical research. We have conducted a lot of studies where we have applied machine learning, and I'm going to highlight some of those. So, for example, we use machine learning to discover drug targets and candidate drugs, and also to predict adverse effects of various anti-cancer drugs. So in one of the examples that I'm going to give you, we were trying to identify drugs that could be used to treat keloids. And we were using multiple data sets, including RNA-seq, proteomics, and histology data. And in another case, we were trying to identify the drug targets that could be used to treat alopecia, hair loss disease, where you have individuals that are very young, balding. And in other instances, we have used machine learning to subtype disease to predict the prognosis of a particular disease and using different molecular data sets. For the project, we are trying to identify drug targets that could be used to treat keloids and the drugs themselves. It took us about 7 years of grappling with that problem, and we didn't get very far. But then we decided to apply AI and bioinformatics analysis, and we're able to identify the candidate drugs and drug targets within a space of 6 months. And then in the project where we're trying to identify the drug targets and the drugs that could be used to treat alopecia, I must state that the traditional pipeline for discovering a drug, once you have the data and the concept, takes approximately 10 years. But in this case, we used machine learning and network analysis to identify the candidate drugs, drug targets, and the biomarkers of the disease within a space of 2 years. So this slide kind of illustrates how transformative AI can be in biomedical research. There are many reasons why we need to invest in research that is at the intersection of AI and microbiome research. For a number of reasons, the key opportunities including that, which has been highlighted a lot in this symposium, is that the African microbiome is quite diverse. And by studying the microbiome in Africans, we'll be able to reach or achieve groundbreaking research that will be relevant for individuals on the African continent. And secondly, is that microbiome data is similar to some of the data sets that I've highlighted to you, for example, RNA-seq data or proteomics data. We're dealing with vast data sets, and that is why machine learning and AI tends to come in for us to identify patterns in the data that we never thought existed and accelerate discoveries in biomedicine. So Africa needs to invest in AI and microbiome research, or research that is at the intersection of these two fields, for many reasons, including the opportunities that will be able to address Africa-specific challenges focused on African individuals and using African data. But when we look at the state of AI research on the African continent, we significantly lag behind in relation to other countries from the other different continents. And for those reasons, there is a pressing need to train many scientists, including public health personnel, biologists, computer scientists, physicists, and among others in AI, and how they can apply AI to their research. And also to strengthen collaborations between African researchers and those in the North, and among researchers from different African countries, and even within the same institution, like researchers from computational biology with researchers from the microbiome interaction lab, so that we can come together and solve some of the most pertinent problems that are afflicting us. And other reasons why Africa needs to invest in microbiome research includes, again, things that have been stressed so much in this symposium. That is the genetic diversity of Africans. Through many studies, we have been able to show, and others have been able to show, that the genomes of Africans tend to vary significantly, like other genomes that are, for example, of individuals of Asian ancestry. And also there are dietary and cultural differences and practices that tend to affect how the microbiome, that affects the microbiome in individuals. And also environmental influences tend to impact on the microbiome. Africa is vast. We've got rainforests and we've got deserts. And it turns out that from those different areas, the microbiome is most likely to be different, as research suggests. And I'm going to highlight this study that was conducted by my colleague a few years ago, where she highlighted the state of microbiome research on the African continent. And she showed that a lot of research in microbiome is taking place in South Africa and some parts of Eastern Africa and a few parts in Western Africa. But I would go further to say that these data sets that we currently have provide us some low-hanging fruits where we can get these data sets and mine them for insights using machine learning and data mining techniques to understand infectious diseases and a lot of metabolic diseases, for example, malnutrition. Overall, I say that the way forward would be that Africa needs to invest in AI and microbiome research, and research that is at the intersection of these two fields, and also including other omics data sets, for example, transcriptomics data and proteomics data sets, for a number of reasons. And the first one is that AI can be used to tailor intervention strategies to individuals, to a population or individuals in a particular region. And secondly, is that AI can help us identify novel microbes, and then we can find interventions or treatment strategies for those microbes. And the third one is that AI is used for predictions. So if you can predict the treatment outcomes, or if you can predict the treatment response or the progression of a particular disease, then we can intervene accordingly and improve the overall public health outcomes of individuals on the African continent. So for this part of our symposium, we have got a list of exciting speakers, and that is the list of speakers that we have today. I'll start by introducing our first speaker. I need to wear my glasses. That blinking case has scorched my eyes, so it's not my friend. So our first speaker is Moses Babibo. So Moses is a professor and chair of the Department of Human Anatomy and Medical Physiology at the University of Nairobi, where he leads research on placental biology and vaginal microbiomes and pregnancy outcomes. His work focuses on understanding the impact of infections, metabolomics during pregnancy, with the goal of improving maternal health outcomes in Kenya and beyond. Moses, please.
Thank you very much, Sinkala. And good afternoon, everyone. This afternoon, I'll be presenting my work, a bit of a very tough title given to me by my friend Joanne. And so I think once this is done, any questions, let's direct them to Joanne. She'll be able to answer this pretty, Joanne is right here. She'll be able to answer them quite well. So my approach to this will be quite generic, but I think we can always translate this information to apply on microbiome work, to women's health, and other areas that are relevant. So AI missed the memo. So how do we unlock the indigenous African data for precision medicine? And of course, I'll invite your contribution in between. So the outline of my talk will be on these specific areas. So where is the African data? How is the landscape of African AI? Then what are some of the opportunities that we have, the challenges, and then how do we unlock this data? And then we have a summary that we can talk about and the way forward. So this, I think, is my most important slide. And if we were to grasp this, then I think my talk will be ended. So we know that these AI rise all over the world. And some of my colleagues, especially in pathology, in interventional medicine, radiology, and other sectors, including surgery, are quite scared. But we know AI is just meant to facilitate, is not meant to replace our jobs. But I see quite a number of people really unsettled by this. But for the AI tools to be reliable, we need very high quality data. We need to have very well-developed algorithms, and that's why we need to work with our computational biologists, computational chemists, computational physicists, and other relevant stakeholders. Then we need to also have infrastructure that can be able to support these systems. And as my colleague, Dr. Sarah, is putting that whatever data we generate has to have a patient-centered or user-centered approach. And of course, data governance and ethics must be integral in this application. So for us in an African setting, that our data has been overlooked when they're developing the kind of AI models that are being used, the large language models that are being used, the neural networks and so forth. Our data, our local data has not been used so much. And we know that without good data, we have wrong evidence. And with wrong evidence, we risk developing wrong AI tools. And therefore, what it means for our population, we are likely to misdiagnose the problems. We are very likely, once we diagnose a problem that's wrong, then we implement a wrong strategy. And with that, our governments, our donors will be able to invest in wrong ways and therefore misdirecting those resources. And of course, the outcomes that we'll achieve will be relevant outcomes. And what it also means is that we cause harm to the people that we treat. So we need to therefore have good data, good AI algorithms that can help our populations. So I'll give a bit of just background on some of the indigenous African data. Is not, of course, holistic, but I think just to give us some context. We know that we have quite a lot of indigenous knowledge and indigenous knowledge systems in Africa. We have our own cultural practices, and we also have a way we look at things. For example, there's a tree known as Mwarubaini. Mwarubaini in Swahili language is Arobaini. Arobaini is 40. In I think most Bantu languages also Arobaini might mean something like 40. And this tree treats about 40 different illnesses. And I just learned it also treats vaginal infections. Okay. It acts as an inflammatory, blood sugar lowering reagent, and antiseptic. We also have this what you call tree of life, the moringa tree. Okay. Who's been touted as when you take the liquid and extract out of that, your immunity is improved. And it's used not only in Africa, but in other parts of the world like Asia, Pakistan, and so forth. We believe quite a lot in spirits, and through communication, some people have received healing, okay? And some of the couples that suffer infertility have had to communicate with the true ancestral spirits and receive some level of healing. But this is our belief, this is our data. And then traditional community healers, meaning that our treatment of disease is just not treating that particular disease, but treating the whole person, treat the psychology, treat their relationships, treat their emotions, and therefore you achieve the whole healing. And these kind of things we always overlook. We have animal-based remedies like children who have bedwetting beyond the expected age, maybe age of 7, the age of 8. Sometimes they are tied with a snake pill. When the snake sloughs, you have the pill, and the baby is tied at the hip with a pill to stop them from bedwetting. And it has worked. I've seen it in my village. For those with erectile dysfunction, they have been fed with the buffalo testes or the bull's testes to improve their virality and their response. These are some of our approaches that we have in our African indigenous systems. However, the Western version of science that is mostly Eurocentric has really led to development of these systems abandoning our African knowledge. And if you look at in large areas beyond health, like economics and so forth, we have this applied across board. So we have a lot of diversity, as been mentioned in this particular room. And because of this diversity and the difference with Eurocentric kind of information that we have, this has affected the kind of decisions that we make for our own populations because we are not using our own data. One of the important elements that I wanted to mention here is that, like during data collection processes, and we are talking to women, want to collect this data from women. Some of our cultures, research done in May 2024 in Nairobi, and the group of focus group discussion of many people say that women who come for data collection in focus group discussion and so forth are not able to give this particular data in presence of their husbands. So because of those cultural barriers, it means the kind of information that you get might not be truly reflective of what women feel in this kind of setups. So the design, the research design, the research implementation has to take into account those kind of differences and the kind of data that we get for application. So what about AI in Africa? What's the landscape like? I've mentioned this, that in many sectors, diagnostics, treatment, surgery, electronic medical records, clerical work in the hospital, AI is really making use and by and large, reducing the cost of care in these particular hospitals. However, there's a lot of disparity between its application in the global north versus where we are today. And mostly the issue, governance structures on how to apply and deploy the AI. And I think this is just not unique in the African setting. It's unique all over the world. But then we probably suffer more of these issues. Then we have little institutional support or capacity. So we don't have very many computational biologists or computational chemists in our institutions. We also do not have so many people aware of how to apply these particular tools. And our governments largely have not been able to place good investment and enough investment to make sure that we use these AI tools appropriately. So the AI is not only used in the health sector, but of course now we're applying it in many other sectors, including farming, smart farming, regenerative agricultural practices, in corporate services, and so forth. And there are quite a number of things that have been mentioned, like the e-learning tools, the analytics, and of course, in the decision support systems where you have chat boxes that can help you make the correct diagnosis or make the correct prescription if a patient needs a certain medication. So as mentioned also, we really still are lagging behind in deploying these AI tools. Some countries in Africa are trying to leapfrog, taking advantage of this, and have developed national AI strategies. And this includes Senegal and Rwanda. However, many other countries are still lagging behind. And about 32 countries in Africa have shown some level of formulating data governance structures and data protection laws, which we know is a very prime thing to do when you're trying to deploy AI so that you're able to protect patients' data and generally the population information. And we now have this concept of Africanization of AI. And that's really, I think, the purpose of this particular talk. How do we make this AI work for us with our African data, and best especially on some of these examples that I've already given, to solve our unique African problems in an African context. So what are these unique opportunities in Africa? This slide, I think, has been overemphasized, and we now know that all over the world, the African population have the highest genetic and phenotypic variation of all the humans, and therefore means that, one, we cannot apply the same tool that we use all over the world for the African population, but number two, the data collected from the African population would easily be useful across the world. Besides that, we have quite a number of unique infectious diseases that afflict Africa specifically. We have diseases like malaria, trichomoniasis, and so forth. And going hand in hand with that is our diverse diets. If you go to North Africa versus South Africa and in between, our kinds of diets and practices of diets are very, very different. And this, of course, affects the kind of microbiome, a kind of metabolism, and a kind of response that we have on various medication. Another important element that I wanted to mention, that oncology and transplant patients, especially renal transplant patients, patients who suffer cancers, for a very long time, we send these patients outside to China, to India, but they're given same medication, same level, the same dose that you get those patients, local patients given in India and so forth. But we found that most of these patients, when they come back after 6 months, you have kidney transplant rejection, or you have this patient dying because of complications of either the medication or the cancer treatment itself. And this is because of the differences in metabolism. We have rapid metabolizers, we have slow metabolizers, and so forth. So it means, therefore, that we need to be able to understand our genome and our drug response and treat our patients accordingly. So in this particular case, we can leverage the use of AI to make sure that we optimize our patients' outcomes. And so how do we unlock this data, African data, to be developed into AI? The first thing that we need to do is that, besides the donor funding and the support we get from our brothers and sisters in the West, we need to also have a lot of investment by African governments in research and development systems that's based and focused on innovation using the African data. And this can also be propagated through a system like the Grand Challenges systems, which some countries in Africa have already deployed. And of course, trying to engage the local communities in traditional medical approach. So we look beyond the Eurocentric version of modern medicine. We also look at how do we approach this condition and how are we able to achieve healing. And you know that as much as Africa contributes to about 15% of the population, we have about 25% of the global disease burden, but we only account also for less than 2% of the genomic data that's known. So over time, the pharma industry and the other centers have really acknowledged this diversity, and they're trying to do something about it so that we have to include the African population in the decision of how these drugs are formulated and what kind of the dosages. But we also need to encourage the African institutions to really participate in clinical trials so that we're able to know what really works for us. So some case examples where AI is being deployed in an African kind of, using African approach and African data systems. Our colleague, Fidel, who is a computational chemist in Cameroon, he's trying to identify the ingredients from traditional medicinal plants to be able to treat some of the unique diseases that we have here in Africa. He's one of our Colestos Juma Fellows colleague. We have some of our researchers who are also trying to use a network neuropharmacology to be able to study what are these extracts from plants, many, many different plants, but trying to come up with the lead compounds that can be able to use to treat issues of formulation of contraceptives that are not hormonally based. So my colleagues from Nigeria as well. And one of our colleagues here, Kelly, posits that use of AI can, of course, be able to really be novel in the use of drug development in the African setup. So some of the challenges that we already mentioned include the issue of data and how we manage our own generated data. What are the ethics? What are the prisons? And how do we share this data across the continents? Of course, the technical barriers that how do we manage and get resources and equipment to be able to do this work? The supply chains, needless to say, we know that whatever else you get, for example, in the United States at $100, here you're probably going to get at $1,000. And this really hinders our research because of these supply chain issues. And then that failed communication, people working in silos, and we not have the government, industry, regulatory bodies, and community leaders communicating and talking together. So in summary, we need to advocate for inclusive AI practices that takes note of our local knowledge. We also need to be able to unlock our indigenous African knowledge in creating solutions that are meant for us, unique solutions for us in Africa. And we also need to encourage collaboration that takes care of indigenous data initiatives as part of development. And my time is up. Thank you very much.
Thank you very much, Moses, for the presentation. So our next presenter is Caitriona Brennan. So Caitriona is a PhD candidate at the University of San Diego, specializing in microbiome research. Her work focuses on using advanced metagenomics and metabolomics techniques to explore microbial communities and their roles in human health. Caitriona.
Can we be seen? Can, of course.
Thank you.
Okay. So thanks very much, everyone. Despite the odd spelling, my name is Caitriona. I'm from Ireland originally, and I'm currently a PhD student at the Knight Lab. My PI, Dr. Rob Knight, will be talking later on. There, I primarily work on method development on the wet lab side of things. And today my talk is going to be about advancing large-scale microbiome research through optimized metagenomic and metabolomic pipelines. So an essential aspect of population-based research is collecting samples outside of a clinical setting. This is crucial because microbial populations are highly dynamic. They vary significantly across time, environments, and hosts, a variability that clinical sample collection alone cannot fully capture. This is why, as we saw from Professor Lebier's fantastic talk earlier on, that at-home sample collection enables the inclusion of a much larger group and more diverse group of participants. And this diversity is so important for capturing the health disparities that are influenced by socioeconomic status and gender, ethnicity, age, and so on. And the first step in actually addressing and reducing these health disparities are identifying them. And that will eventually then lead to reducing the health inequalities we see today. So late this year, myself and a bunch of collaborators, we performed a literature review on how manipulating the microbiome can enhance female health. We focused on 5 key methods: microbial transplants, probiotics, prebiotics, diet, and synthetically engineered microbial strains. And despite the limited number of studies, there is clearly a potential for targeted microbiome interventions that could revolutionize the management and treatment of common yet challenging conditions. And while many of these findings are promising, many studies lacked appropriate sample size and diverse subject groups. And there are even instances where separate studies had conflicting results. This highlights the fact that our understanding of microbiome dynamics in the female health and reproductive disorders is still in its infancy. And as we learned over the last few days, that is going to change very quickly because of a lot of the wonderful people in this room. But I'm going to talk about some of the aspects that maybe do hinder microbiome studies. There are many barriers, and the most common of which include insufficient sample size, which is connected to the per sample processing cost, also connected to funding issues, lack of standardization across methods, making meta-analysis difficult, inadequate preservation that incorporates bias, cross-contamination, which especially hinders low biomass studies, human error during sample accession and metadata curation, and low-resolution sequencing techniques that limit our ability to detect pathogenic strains. So, one of the most important aspects of microbiome studies, I believe, is sample accession, because if this is not done correctly, it leads to unresolvable issues downstream. On the left here, we see the loading of a cargo ship in 1945 with various size shipments creating chaos, delays, and often resulting in lost or damaged goods. On the right, then, we see a stark contrast where containers of a standardized size are efficiently loaded onto a ship using a crane. And these uniform containers can be neatly stacked, saving time, preventing loss and damaged goods. And this actually resulted in a 10-fold cost reduction compared to the method on the left. So this perfectly resonates with sample collection as we see today, where we often encounter tubes of various shapes and sizes, sometimes coupled with illegible handwriting. And this perfectly mirrors the scenario that we saw from 1945. On the other hand, then, adopting standardized barcoded 1 ml tubes is akin to the modern cargo loading method. These standardized tubes can seamlessly assemble into racks suitable for automation, streamlining processes and ensuring the integrity of our collected samples. These barcoded tubes in particular are available from Thermo Fisher, and they form an integral part of our sample accession, DNA extraction, and metabolite extraction pipeline that we have coined as the matrix method. So previously to the implementation of the matrix method, we used to collect samples for DNA and metabolite extraction on 2 separate swabs. These swabs would be added to tubes, which we would attach barcodes to and scan individually as they came into the lab. We would assign then these barcodes to a plate map, where each sample would be assigned to a well on a 96 well plate for both metabolomics and metagenomics. And we'd have to break each one of these swabs off into these wells, which you can imagine is extremely tedious and very error prone. Also with these plates during the lysis, the bead beading step, it is very prone to cross-contamination because of all the wells on these plates share a single seal. On the other hand, then, the matrix method requires just 1 swab for both metabolomics and metagenomics. And these swabs are made from high-density polypropylene. They don't leach, which would cause an upset for the metabolomic side of things. And when they come into the lab, they're actually pre-filled, though, with 95% ethanol. And this 95% ethanol works as a storage solution, keeping the microbial communities preserved, which we have demonstrated in a previous publication. So this really is very cost effective because it enables the shipment at room temperature without the need for dry ice. So once we have a full rack then of these tubes, we can scan them using a high-speed barcode reader, which is also available from Thermo Fisher, and this can scan them all simultaneously. So within a couple of minutes, you're ready for downstream processing, unlike 4 hours compared to the other method. And here's just a quick film of the barcode reader in action. As you can see, it can scan things very, very easily. Could you imagine scanning each one of these tubes using a handheld barcode reader? It would take you much, much longer. And you can download a CSV file here and then upload it into your metadata very easily. And just to further highlight the importance of lab hardware for these pipelines, we have what's called a cap-it-all, which caps and decaps tubes. Seems like a very, very simple task, right? But could you imagine uncapping all of these tubes by hand? I know for me anyway, it would take me over 5 minutes and probably involve caps flying all over the bench. So this is very, very important. There's also a cheaper alternative to this where it's like a handheld one with 8-channel. So that's the sample accession side of things. And I'm going to fly through this for the sake of time, but this is the extraction part, where once we have a full rack, we add it to a plate shaker to actually so here, the 95% ethanol, it not only acts as a preservative, but it also acts as a solvent for the metabolomics. So we shake the sample, we centrifuge it to separate the metabolites, and then we take the supernatant off using an 8-channel pipette into a deep well plate suitable for metabolomics. With some further processing, then it's sent off for mass spec analysis. Then we're left with the pellet, with the microbial communities and the DNA. We add it to a speed vac to get rid of that excess ethanol, which would interfere with downstream analysis. We add the lysis speeds and the lysis buffer. We perform bead beading. And then similar to step 4, we transfer the supernatant into a well suitable for the Kingfisher, which is the DNA purification system. So the DNA extraction method that we benchmarked the matrix method to was the MagMax microbiome kit. It's routinely used by labs, and it proved to be superior to the other 5 kits tested in this publication. Okay, so to assess well-to-well contamination across the 2 methods, 4 of our lab techs independently conducted each method in duplicate. 3 technical replicates of human fecal swabs were obtained from 4 volunteers, and each fecal swab was meticulously transferred to corresponding wells of the deep well plate from the old method, and then the matrix tube rack from the matrix method in the same positions, here denoted by the brown circles. And then these were surrounded by 84 blanks. The high amount of blanks will give us an indication into the positions and the distances that well-to-well contamination can occur. So you can see here from the plate-based method, there was a much higher level of contamination, with 128 blanks actually being contaminated out of 672, with an average concentration of 0.21 nanograms per microliter. On the other hand, then, the matrix method only resulted in 14 out of the 672 blanks showing with contamination, with an average of much lower 0.026 nanograms per microliter. So a dramatic reduction in well-to-well contamination during the extraction. Untargeted metabolomics, then, it's frequently performed on human fecal samples in conjunction with sequencing to unravel gut microbiome functionality. Therefore, metabolomics experiments, they should adopt to accommodate the safety and needs of at-home collections. The typical solvent for extracting metabolites is methanol, but this is not suitable for at-home safe sample collection. Therefore, we also compared metabolite extraction using the matrix method to commonly used and commonly validated methanol extraction. So these PCA plots revealed that even though the samples clustered according to the extraction type, the influence of subject is much greater, as shown by Permanova here. And this upset plot displays that more than 75% of the obtained features are recovered by both extraction methods, encompassing 94% of the total annotated features, with more features being captured by the matrix method. So we also then had to look at, you know, is the metabolome stable at room temperature if we're going to be shipping it in 95% ethanol? And indeed, it is relatively stable. So you can see here that these subjects, so each one of these points, by the way, is a sample, and you can see that they cluster quite tightly on a per subject basis, with very little deviations according to storage time. And again, this upset plot displays that more than 88% of the obtained features are recovered via both extraction methods. And these include 94% of the total annotated features. And you can read more about the matrix method in our recent publication on M systems. There's an in-depth metabolomics publication incoming, which was actually posted on bioRxiv yesterday. So with that, the matrix method does address to some degree these 5 pitfalls, let's say. And so I'm just going to go through this very quickly. Shotgun metagenomics has become a very popular tool in microbiome field due to its ability to sequence whole genomes. But there are some privacy concerns coming up now with shotgun metagenomics because of host DNA leaking into microbial data sets. And also assemblies are very difficult to form from shotgun data. As you can see, here's a typical assembly from shotgun data. It's like trying to put together identical puzzle pieces. So you can see it's a complete mess. On the other hand, long read sequencing, this can lead to improved taxonomic resolution, enhanced genome assembly, the detection of novel species, and an insight into mobile genetic elements. We initially avoided PacBio because of its low throughput and high input requirements. However, recently PacBio have come out with the new sequencer called the Revio, as shown here. This is the first one that was ever launched on the African continent. It's located in Pretoria. And what's really good about PacBio sequencing is no PCR bias. It has an accuracy on par with Illumina's short read sequencing. And also, we compared fecal samples that were processed using TELSEQ, Shotgun, Oxford Nanopore, and PacBio. And we can see that PacBio actually resulted in much higher quality and longer contigs compared to the other 3 methods, which will then lead to much higher quality assemblies, giving you much stronger resolution into what's in your sample. And with that, I'll just talk about a quick potential challenge that we have is, of course, the low biomass. What do we do with them? Where we're going to test a beta kit here that will amplify the microbial DNA, and hopefully we'll be able to reach the input requirements for PacBio. So with that, we address all, to some degree, all of these main pitfalls, and hopefully we'll be able to enable home sample collection and much more cost-effective shipping. So I'd like to say thanks very much to everyone, especially those back in the night lab for all their help, and my PI, Rob Knight, who will be giving a talk later. Thanks very much.
Thank you very much, Caitriona, for the nice talk. I love that you decided to correct my pronunciation of your name right on the first slide. A cat and a tree, followed by na. You're used to it. Yeah. So I'm going to move on to our next speaker, who is Karl Burgess. Karl is a chair of biological mass spectrometry and associate dean of impact for the College of Science and Engineering at the University of Edinburgh. He is originally from Glasgow, but he told me not to tell you that. I'm sorry, Karl, I've told them. He's particularly interested in developing metabolomics tools to support biotechnology, medicine, as well as global partnerships to generate impact. Karl, please.
See if I can get this working.
So I am not an epidemiologist or a microbiome researcher, I'll tell you that to start with. Oh, this one. Yeah. It might be easier so I can walk around. No, that's fine. Thanks very much. Can you hear me okay? Cool. So, as I said, I'm not a microbiome researcher much. I'm a mass spec geek, as you can tell from my bio. So I'm going to talk mostly about a single paper. It's very reductionist. And I would like to thank the guys this morning for giving a really nice segue into this. So what I mostly work on is biotechnology and developing new techniques for biotechnology and analyzing biotechnological processes. And if we're looking at the things that we need to treat the microbiome, these are all things that if we want to actually get them into clinical use, are going to need to be scaled up. And the way to scale those up is by using biotechnology. And most drugs going through approval at the moment are either things like cell therapies, where you need to get large biomasses, or you're making protein drugs like endolysins, or even small molecules like antibiotics and things like that are also made using biotech. Most of my group, I mostly put this up because it's Jess's birthday today, and that might annoy her. But most of my group works on this system, which is this real-time metabolomics thing that we've developed. So basically, this is a bioreactor growing anything from E. coli to yeast to Chinese hamster ovary cells, producing monoclonal antibodies. And this is coupled up directly to a mass spectrometer. So during the full course of a fermentation or a bioprocess, we can get a sample every 5 minutes. So currently over a 14-day fermentation with CHO cells, we can get over 2,000 samples. With a full metabolome for each one, which gives us this really good idea of what the biochemistry of the system is happening. And regardless of whether you're making biomass, a small molecule, or a protein, these are metabolic processes. And so metabolomics is really important for detecting these. So we have just kicked off a microbiome manufacturing product for the gut microbiome, but I'm not going to talk about that today, but that's Jess's project. What I am going to talk about today is surfactant production in bacillus. So surfactant is a surfactant, sort of a soapy thing. And it's interesting because it's a high-value lipopeptide with surfactant and antibiotic activity. And one of the interesting things about this is it's a non-ribosomal peptide. So it's a biosynthetic gene cluster from the fatty acid and amino acids pool. If you're doing synthetic biology mostly, what you want to do is a nice long chain where it works like a pipeline, and you put your precursors in and you get your product out at the end. This does not work like this. So most of the standard methods for optimizing it don't really work very well because you have to optimize basically everything, like lipid production, all the way to amino acid production. So that's really tricky. So where this came from, the idea for the project was mass specs are really, really expensive and really annoying because they break down all the time. And so what we want to do is maximize the time that we use for them. And to do that, we want to reduce the cost per sample. And so we want to make the samples really, really fast. And so a lot of work that I did since I moved to Edinburgh from Glasgow was based around making really fast metabolomics methods. So the current platform that Tessa runs uses as a standard a 4-minute method, which gives us a really full metabolome. But we wanted to push this further. And because we know kind of what we're looking for in this system, we started using a flow injection method that takes a minute per sample. So we can really blast through samples. And so we wanted to use machine learning as a way of doing experimental design and helping us improve this system to increase yield. So to do that, when I moved to Edinburgh, when I moved to Edinburgh, I ended up with Diego as a roommate. He's a fantastic guy, and he's a machine learning expert and a computing scientist. So we had a lot of conversations to see how we could interact. And we ended up with this project and this really, really great PhD student called Ricardo Valencia Albornoz, who submitted his thesis on Monday. So I'm very pleased. Not that I'm pleased that he's leaving my lab, but I'm very pleased that he's submitted it. But this system is really difficult to get high yields, as I've previously mentioned. So what are we going to do? We're not going to do genetic engineering approaches just because it's complicated and fiddly. We're just going to mess around with the medium and see what we can do by just changing the medium that the cells are grown in. So we're using M9 as a base, and we're using the surfactant C variant. So it actually produces a few different soapy molecules, just different structural arrangements of these molecules. And so all we're going to do is we're going to change the glucose and then the ammonium, because you need both nitrogen and carbon to make amino acids. So can we get sample efficient optimization and rich metabolomic data from the same pipeline, right? So we're going to do the optimization cycle, and we're going to collect metabolomics data while we're doing it. So this is the pipeline that we ended up with. So what we get it to do is we say, okay, computer, I want you to tell me what the starting conditions to optimize on are. And so what the algorithm does is it says, right, here are the conditions that you want to try first. We're going to try and get a yield optimization surface out of this, which I'll show you in a minute. And then that builds a machine learning model. It uses Gaussian process regression on the data and the yield measurements. So we're getting the output using metabolomics, but we're also getting all the other metabolomics data as well. And then we say to the algorithm, right, what are the next set of samples that we need to take to find out what the optimum is? And then it goes around the cycle again. So we chose a variety of different starting conditions and told it to go ahead. Of course, we're generating very large numbers of samples with this. So Ricardo disappeared for a month or two, and I discovered later on that what he'd actually been doing was he'd borrowed a robot, a liquid handling robot or somebody else, built his own robot arm, and used that to move plates around and things. And he was using some bioreactors as well to just bulk up his bacterial colonies and things. So that was cool. And this is the sort of starting data for the results. So we were taking OD measurements over time, so we could see the bacterial growth, and we were seeing what the yields were in terms of the surfactant from the mass spec. And this is the sort of data that we're getting. So this is the yield surface. So these dark blue patches show, sorry, dark green patches. I'm not colorblind, I just can't speak. These dark green patches are bits where you've got very high yield, and the white patches are where you've got low yield. And on this side, which I think is one of the cool things about this Gaussian process regression, is that we get a measure of the uncertainty of the measurements as well. So these are the first combinations of feedstocks that we started with. And these are the bits where you have good confidence, and the blue bits between them are the bits where you have low confidence in the measurements, right? So you've got this idea of good quality data and bad quality data, which I like from this technique. So that's the first iteration. And then the active learning algorithm said, right, it can see that there's a high yield here. I want you to take a better look at that area specifically. So we then did that, and we then have a completely different sort of pattern of certainty in the measurement and the overall yield surface. And you can see that the yield is massively increased. And we also get the OD measurement and things like that. And then the final one, it says, okay, I want to look at the edges of this to try and reduce the uncertainty there. So it's then pushing out towards the edges in terms of the certainty of the predictions of this whole system, as well as the type of data that we're generating, so that we can end up with a good measure of the best possible combination of medium that we can do for this. So what we got out of this was a 60% increase in overall yield from baseline, which I thought was pretty good. So after that, we're increasing the number of factors and all this kind of stuff. So we've got multiple factors, not just glucose and ammonium. We can do 10 factors at a time, and we can use lots of different optimizations. So not just yield, we can look at lots of different optimization things. So, but along with that, we get all the metabolism as well. So not just the inputs and the outputs, so the glucose, the ammonium, and your yield, but we can also have a look at the metabolic state of the organism, which I think is really nice. And you get the same maps and the same measurement confidence data. So to extrapolate this beyond biotechnology, we're looking at this system where you can get massive amounts of information about the metabolic state and how that metabolic state changes depending on outcomes. And those outcomes could be anything. They could be disease states, they could be whatever. And the inputs can also be any sort of parameter that you care to put in a system. So I was really excited by this because we ended up with this kind of concept of metabolic anisotropy, which we published in the paper. And it's basically if you've got a surface like this and you've got two factors that you're varying, the impact on that surface will change really dramatically or not dramatically, depending on which way you walk effectively on that surface. Which seemed kind of obvious, but it's quite an exciting idea because what you can do then is you can go, okay, I'm stepping over here. How does that affect not just the outcome and the inputs, but the whole metabolic system from that? So we're trying to follow this further with a couple of grants at the moment, but I think it's quite an interesting idea of what we can use as an overall model of metabolism that you can get very, very cheaply in a minute's worth of run from any biological system that you care to mention. And you can predict using this completely empirically what happens if you shift it in a particular direction. And you can just see from here, if you start from the top here, these are the sort of directions that you can move on this map of just glucose and ammonium and the impact that it has on various different metabolites, all of which are central metabolism, the key things that we share from the human organisms all the way to bacteria. So I think that's quite cool. And then we can fill in the gaps as well and predict these Pareto fronts, which basically give us the trade-off between optimization surfaces. So in this particular case, there's not much of a trade-off. So this curve here, so the best possible outcome is somewhere here. So there's not a massive difference in this area here. Whereas if you look at a possible different surfactant product, this Caitriona, there's a big trade-off. So there's if you're trying to improve the number of bugs versus the outcome, you have to make sacrifices to do that, which I think is quite an interesting outcome. So I've underrun a bit, that's fine. I don't know how this has ended up like this, but so bioprocesses are very complex living systems, and they're sensitive to many parameters. I think that's completely understandable. We've talked about diversity an awful lot, and diversity is absolutely critical in mapping metabolism as well. I think in the microbiome, this is something that's absolutely critical because metabolism is critical in the microbiome. All of these bacteria communicate with each other. This whole system is just a massive interconnected group of different organisms shouting at each other chemically all the time, right? And I think that's a really exciting area. But the only way we're going to be able to understand it is by parameterizing this and using these very large-scale metabolic analyses, as well as genomics, to really tease out this data. We can get one of these cycles in less than a week. So if you're trying to optimize a bioprocess, what would be typically a PhD student project to optimize medium, because I've run a couple previously where it took an entire PhD to generate a new medium. We can do this process in about 3 weeks with 3 iterations with the robotics and all that sort of stuff, which is pretty cool. And I think that the future of bioprocessing is a better understanding of the link between process parameters and outcomes. But I also think that the future of metabolomics is a better interconnection and understanding between all the different factors and understanding the models. And I think that's not something that we do particularly well at the moment. And I think that's an area where machine learning and artificial intelligence can really help. So I will finish there. I just want to thank the Medical Research Foundation, who paid for me to come down here and spend some time with Tarek, which is fantastic, and also gave me the opportunity to see all of you guys, which is great. And it's a real privilege to be here. And the people who worked on this, particularly including Tessa, who's our core facility manager, who's in the audience, and please give her a shout if you're interested in talking about metabolomics. So thank you very much.
Thank you very much, Karl, from Edinburgh. So our next speaker is going to join us online. His name is Sean Gibbons. So Sean is an associate professor at the Institute of Systems Biology and an affiliate associate professor of bioengineering and genome science at the University of Washington. His research focuses on ecology and evolution of microbial communities and their impact on human health.
Hi, everybody. I actually can't share my screen, so can the AV team give me permission? Do you hear me?
Yes.
Okay, here we go. Now we are working. Okay. Can everybody see the correct screen? Yes? I'll assume yes. All right. Thanks, everybody. It's very early here, so I'm still getting my bearings about me. But it's a real pleasure to be with you. I am sorry I can't be there in person. Some grant deadlines I just couldn't avoid. But I'm honored to be here to speak with you. And I really enjoyed the last couple of talks that I was able to hazily catch this morning. So today I'm going to talk about a kind of modeling, not necessarily the traditional artificial intelligence that we've been hearing about. This is mechanistic modeling. And how far can we push mechanistic modeling to be predictive in microbiome science? So as some background, we've known for a couple of decades now that the microbiome is a modulator of our phenotype for hundreds of different phenotypes that have been tested in germ-free animal models. This is a particular experiment that Peter Turnbaugh did a while back, where he took feces from obese and lean folks, humans, and transplanted that feces via FMT into germ-free mice, fed them the same exact diet, and the mice that received the transplant from the obese individuals gained more weight on the same exact diet, right? So the microbiome is key to this particular response, but to many, many others as well. However, despite the fact that we can see this causality and we know that this is going on, it's been a little difficult to translate some of this to humans and to healthcare, because often we don't know the mechanism for exactly what's going on, what is mediating these phenotypic transformations. So the last talk set this up very well, but I would argue that one of the key mediators of function in the microbiome is metabolic crosstalk between the microbiota and the body and the host. We know that our microbiota, our gut microbiome, is like a anaerobic bioreactor sitting down in our bellies. It takes in myriad dietary substrates in addition to host substrates like mucins and bile acids, turns them into another large subset of bioactive compounds that are absorbed into the bloodstream and can travel throughout the body and affect distal organs like the kidneys, the liver, the brain. And recent work from our group from a couple years ago, I guess now, showed that about I think it was 44% of all circulating metabolites in the blood that we measured via untargeted metabolomics showed some significant association with cross-sectional variation in the composition of the gut microbiome. And that was much stronger effect than we saw for host genomic variation in a population of about 2,000 folks. But the genome was associated with about 10% of circulating metabolites. And so what we really need in order to harness this process is to be able to integrate all of this information and make predictions, right? Everyone's got a unique composition. We all have totally different microbiomes, different sets of genes, 100-fold, 150-fold more genes in our microbiome than there is in our genome. And how do we integrate all this information? We need some sort of Rosetta Stone that can decipher this conversation that's occurring between ourselves and our microbiota in this language of small molecules. So how do we integrate complexity? There's research from back in I think the 50s. Psychologists found that a human being can hold about 7 things in their head at once, right? So as soon as you get above 7 variables, you cannot hold them all in your mind. We need some sort of externalization to integrate this type of complexity. And the latest buzzword is artificial intelligence. It's a type of statistical model, usually, is what people are referring to, as a way to integrate lots of information and make prediction. And like I said, the version of artificial intelligence that's usually discussed today centers around statistical AI or deep learning, neural nets, and you'll be familiar with something like ChatGPT, right? Which this is an output of here, but also other generative AI like image generators and things like this. These are models that are trained on large corpuses of data and are trying to extract information to make prediction from those corpuses. However, if you go back to the early days of artificial intelligence, the original AI was actually what's called symbolic AI, which is just hard coding in a model of the universe as you understand it. And so this is an example of just self-limiting process, differential equation of growth. But Newton's laws of mechanics could also be a formalization of a symbolic model. And often in biomedicine, when we talk about symbolic systems or symbolic AI, we're using some sort of knowledge graph, which is a list of interactions between entities that is sort of validated experimentally, like enzymes using particular substrates and producing particular products. So as an example of the statistical AI side of things and how that's been deployed in the microbiome space, there's this really charismatic example from 2015, Zivi et al., that many of you are probably familiar with. And essentially what they did here is they had a population of, I think, 800 to 900 folks, where they collected a bunch of dense phenotypic information, including their dietary intake, clinical chemistries, microbiome sequencing, in addition to continuous blood glucose monitoring. And they trained, I think it was a gradient boosted decision tree kind of model to predict this glucose response from the underlying diet and the microbiome composition, along with some of these clinical chemistries. And they got a pretty good prediction. So they found that they could essentially predict optimal diets and suboptimal diets for these various participants, and that these optimal and suboptimal diets performed competitively against nutritionist designed dietary interventions. And then subsequently, this particular research group tested their precision model against a standard of care dietary intervention, which in this case is the Mediterranean diet, or MED. And they found that they could actually improve post-meal glucose response better than the Mediterranean diet could do for the majority of people in their cohort. So this is sort of a nice proof of concept that their precision model was actually working. There's a company, Day 2, that's kind of commercialized this, although I believe they've just recently wound down operations, sadly. And then Zoe is another one of these companies that's using ML models to make precision nutrition predictions. But our group has been taking a bit of a different tack here. So rather than using machine learning to make nutritional predictions, we're trying to leverage genome scale metabolic modeling, which is essentially a form of symbolic or mechanistic artificial intelligence. And I'll get into that in a little bit, but this big hairball here is just a picture. It's not supposed to be informative, but it's just a kind of pretty picture of what one of these community scale metabolic models looks like that we're building, where each organism, each species in the system is essentially one of these regions of high density. And you can see all the internal metabolic fluxes that are going on inside of a cell. You barely see, lines across these regions, which are cross-feeding interactions between organisms. And then there are kind of larger swirls of extracellular metabolism occurring outside of the cells that kind of is shared across the community. These are very big models with tens of thousands of reactions and metabolites and hundreds of thousands of connections. And we've built a tool to enable the construction of these types of models. So classical flux balance analysis is usually applied to a single organism, like E. coli growing in a bioreactor, or even small consortia with, up to 5 or 6 species. But as soon as you get to very diverse systems like you find in the human gut, these methods can break, and they can be actually very time consuming as well to run. So we've put a lot of effort into optimizing both the speed and the accuracy of these types of models. And I should say this platform, MyCom, which is a portmanteau of microbial and community, was designed by Christian Diener, who was this super talented research scientist I had in my lab for several years. Now he runs his own group at the Medical University of Graz in Austria. So basically, I don't have a lot of time to go into the details of the modeling, but what you need in order to initialize sort of a personalized metabolic model of someone's gut microbiome is compositional information. So you sequence their microbiome with metagenomics or even 16S, and you can see taxonomically who's there. You can map those IDs to a database of metabolic models like Agora, and then pull down genome scale reconstructions of the metabolism of each of these organisms, and that initializes this composition. You also need a dietary input for the modeling. We have several pre-computed diets, like the standard Austrian diet. I don't know why we have the Austrian one, we just do, and then the keto diet and the Mediterranean diet and a few others. But it's possible to build personalized diets as inputs as well. If you have metabolomic data or other types of data that can be used, I guess questionnaire data is probably the easiest to be used to construct this dietary input. We do some tricks here to make the diet more like what it would be experienced as in the colonic milieu, where we strip all the metabolites that are known to be absorbed in the small intestine using the Recon 3D model of human metabolism. And then we add in a few metabolites that we know the host is injecting into the diet, like mucin and bile acids. As an output, we get predictions of growth rates of all the taxa in the system. And then we get all of the cross-feeding interactions, the net consumption of metabolites from the medium environment or the diet, and the net production of metabolites. And we call these things microbial community scale metabolic models. These are purely mechanistic models. We're not fitting these models statistically. They're just mechanistic models of an individual person's microbiota, constrained with their personalized information. And so what is flux? I'll just quickly brush over that now. We can measure function in microbial ecosystems in several ways. You can sequence the genes, you can measure the proteins, you can actually take snapshot measurements of the metabolites with metabolomics. But these snapshot measures may not always be perfectly informative, right? A metabolite may be present at a very low concentration in a given snapshot, but it might be produced at an incredibly high rate and consumed at an incredibly high rate. So it might be a central node in the entire process, and you wouldn't understand that unless you had a dynamic picture of the actual flow of mass and material through the metabolic network, which is flux. And this flux is exactly what these FBA models are predicting. So I'm going to pivot quickly to one particular validation of our microbial community scale metabolic modeling platform in terms of butyrate production. And so why do we choose butyrate as one of our first targets to look at in terms of what is the predictive accuracy of these models? It's a really, I would call almost a drug. It's a really potent cardiometabolic drug that is produced endogenously in the microbiota. It's only our microbes that are making it. We don't make butyrate, and we are underexposed to butyrate in the modern world, at least in developed countries, especially because, if you look at the American diet, we're maybe eating about 10 grams of fiber per day. Whereas if you look back to kind of hunter-gatherer lifestyles, we were probably consuming closer to 50 to 100 grams of dietary fiber per day. And that fiber is fermented into organic acids like acetate, butyrate, and propionate. And so our exposure to butyrate is much lower than it used to be. And butyrate has effects across the entire body. I won't go into all of them, but it affects host receptors. So it's an HDAC inhibitor. It actually inhibits epigenetic decoration of the genome. It can bias T cell differentiation to Tregs and reduce systemic inflammation in the body. It can also bind to receptors in enteroendocrine cells, which can stimulate production of hormones like GLP-1, the appetite hormone that's talked about a lot with these GLP-1 agonist drugs that are on the market. And so its effects are widespread throughout the body, but generally higher butyrate is better. It improves cardiometabolic health, reduces your chances of type 2 diabetes and heart attacks and so on. And so this validation work was recently published in Nature Microbiology, and we were very fortunate to get the cover in the July issue. And so you can kind of see that work there if you have more questions. But this particular work was driven by Nick Quinn Bowman, a graduate student in the lab who just graduated a couple months ago, along with Kat Ramos and Christian Diener, who helped with running the experimental validations. And so how do we validate butyrate flux? So this is actually tricky because butyrate, it's produced in the lumen of the gut, and almost as soon as it's produced, it is absorbed by the intestinal epithelium and it is burned, right? It is converted into biomass and CO2. The intestinal epithelium uses butyrate as a preferred carbon source and energy source. over even blood glucose. And so it may be produced at a really high rate, but if you measure it in stool or if you measure it in blood, it can be really hard to catch or see because of this issue. And so in order to correctly measure butyrate, this is my cat Oscar, by the way, he got me at home today, we have to take poop out of the body into our anaerobic chamber, and we run these what we're calling poop soup experiments, but if we're being more serious, we call them ex vivos. And we do really short-term time series incubations of stool with various additions, macronutrient additions. So we have a control where we say add PBS buffer only versus a dietary fiber. We incubate over time. We take measures of metabolomics targeted across time. We can use that information to calculate a flux, right? The net production of these short chain fatty acids over time. So when we ask, how does our model predict these fluxes, right? We can run the same exact intervention in silico in these models as we run in vitro, and we find, decent prediction. We weren't actually expecting to see this. Mechanistic models classically aren't always the most predictive kinds of models, but our R values are about 0.61, right? Or, like 35 to 40% of variance explained generally is what we find. And we were pretty happy with that. We weren't quite sure how to compare that to what's been done before. So we looked back at that Zevi et al. Paper that I talked about earlier. And this is one of the main panels from, I think, their figure 3. So it's essentially the blood glucose predictions, the post-meal blood glucose response predictions versus measurement across this population, and they get an R value of about 0.68. So a little better than what we're doing, but not too much different. And this is a model that's been fit to data, right? So here there's no fitting, and we're getting kind of on par levels of prediction. And of course, this prediction actually led to a translational outcome in this day 2 company. So we're bullish about the predictive capacity of these MCMMs. So the next set of validations we ran was to look at the relationship between these predictions and measurements of these clinical chemistries, health markers in the blood. So we had a population of about 3,000 people with paired microbiome data and blood clinical chemistries, and we see sort of what we expect. So the middle column here is the predicted butyrate production flux from our models. We see a lot of significant associations, which are these stars. For propionate predictions, we don't really see much associated with blood measures of health markers. And for blood measures of butyrates, we have metabolomic measures of butyrate in blood, fasting blood, and we see really no association. So blood butyrates like white noise. The predictions give us good coherence with the data, and the directionality of this is what we expect. So markers of lipid health like LDL cholesterol and triglycerides are lower if you produce more butyrate. Insulin resistance markers are lower or better when you produce more butyrate, and levels of inflammation are reduced when you are a higher butyrate producer. Great. So assuming that the modeling works, I haven't put together the funds yet to run a human clinical trial, but we can run a sort of thought experiment with in silico simulations. So taking this same population we looked at for clinical chemistries, we can flip them between 2 different diets in silico. The standard European diet, which I think is based on the Austrian diet, it's fairly similar to the American standard diet, and a high fiber diet. When we do that, we find that there are some people who are non-responders. They don't produce much butyrate when you flip them to a high fiber diet, and some are even regressors. They actually decline a little bit in their butyrate production. And we asked if we could, rescue these non-responders and regressors with simple co-interventions on these background diets, either prebiotic fibers, inulin or pectin, or Faecalibacterium presnitzii, which is a butyrate-producing Clostridium. And so when we look at butyrate production, when we flip between diets, yes, we see a big increase when you go to high fiber. But again, there are some people down here in the tail, because of this high variance, that aren't that do not benefit from this dietary intervention. And so looking at personalized co-interventions on top of these diets, this is what I'm plotting now. The heat now is butyrate production in red. The non-responders are in green. The regressors are in yellow. The rows are dietary interventions, and the columns are individual people. And each of these dark boxes is the optimal intervention for that one person. And if you look across this plot, there is no single intervention that does best for everybody, right? For some people, taking a prebiotic fiber like inulin really turns up your butyrate production from the baseline. Whereas for others, it doesn't matter how much feedstock, how much fiber you feed down their system. If they don't have the genetic capacity to produce butyrate, they're not going to make it. So they probably need some sort of probiotic intervention. And that's often the case for these non-responders. They seem to kind of lack the capacity to produce butyrate. And so this is the initial proof of concept for this. We want to try to apply this type of modeling framework to a lot of other questions, like engraftment of taxa in the system, predictions of other metabolites. We're still generating data to validate those predictions. And with that, I'll stop. I'll thank the members of my group, the Micronauts here, for all their great work on this, for the funders. And I'll quickly end by saying, with these mechanistic models, there is an advantage to them as well, in that because we're not fitting to a training cohort, the applicability of the models is a little more uniform across cohorts, right? So we're not overfitting to, say, a European population or an African population or an Asian population. The models should be a little more robust to cohort variability, with the caveat that our database of models, the types of organisms that are represented in those models, are depleted in the types of taxa that are found in populations outside of the US and Europe. So more work is needed to improve our databases. And with that, I thank you for your time.
Thank you very much, Sean, for the awesome talk. Could you please remain on the call until the end of the next presentation? Then there will be a time for questions. Thank you. So our next presenter is going to join us online from the United States of America. His name is Rob Knight. Rob is a computational microbiologist and a professor at the University of California, San Diego. His research focuses on developing techniques to characterize microbiomes in humans, animals, and the environment. Rob will be giving us a talk entitled Addressing the Missing African Microbiome. Thank you very much, Rob.
All right, great, thank you. Let me just stop screen sharing. All right, can you see that okay?
They are still loading your slides.
Can you hear me?
Yes.
Okay, great. So can you see my slides?
Sure.
All right, excellent.
You can go ahead.
So good afternoon. This African Microbiomes and Health and Disease Symposium is a very important effort, and I'd also like to congratulate you on the forming of the VMRC. I'd also like to thank Joanne, Anna, Lenine, and Heather for inviting me to speak, and for hosting Caitriona in person. I wish I could have been there in person myself, but I had to be in DC for induction into the National Academy of Engineering, so I'm doing this virtually. And it's been a wonderful meeting so far, so congratulations on that. I'm going to start with acknowledgements, just to make sure that we just make sure that we get to them on time. So the work that I'm going to show you is a combination of work in my lab, in Peter Dorrestein's lab, and at the Center for Microbiome Innovation. And you heard from Caitriona already in her excellent talk about some of the lab methods that we're using to produce AI data at scale. For this talk, Joanne asked me to comment more on how that will be used downstream, and what some of the challenges and opportunities are for application in Africa. I do have some relationships with the companies that I'm required to disclose. None of these are particularly relevant to the current talk. So the reason the reason we're excited about studying complex microbiomes is that microbes are intimately involved in many of Earth's problems, and these include infectious disease, chronic disease, including psychiatric disorders such as major depressive disorder, which you might not have thought was linked to the microbiome, but actually is. And then outside the human body, food production, pollution and responses and resilience to pollution, and climate change. And harnessing microbes to address these problems requires both better data than we have currently and also better quantitative approaches. And these have been very much a focus of my lab's research over the past 20 years. Unfortunately, although the problems that we need to address with microbes are global problems, the data sets that we have available are very biased geographically. And this news article came out in Science in 2022, basically just documenting where human microbiome samples are from around the globe. And what you can see is that essentially all of them are from Europe, Canada, and the US. There's some from Eastern and Southeast Asia, and there's very little from Sub-Saharan Africa, even less from Northern Africa. And as I commented at the time, this distribution of samples really speaks to deep inequities in how research is funded and conducted. And so, if we're going to be able to use microbiomes globally, especially to address health problems, we need a much richer, more representative training set. So, in our own work, to increase microbiome representation, we've turned to crowdsourcing and crowdfunding in projects like the Earth Microbiome Project, the American Gut Project, which has now become the MicroZeta Initiative, and others. And this has been quite useful for increasing microbiome representation generally, but I have to admit that it has not worked for Africa, at least as done so far. So I'll show you some illustrations of what we can do. What I'm showing you here is an animation of successive sequencing runs from the American Gut Project, and the different points are colored by different sites in the body. So the stool samples, which we have the most of, are in red, and then the oral samples are in blue. The skin samples are in orange. And what you can see is that as we accumulate thousands and thousands of samples over the successive sequencing runs, the population fills in more and more densely and in the same locations. So you can see that with good process control and quality control, which we continue to elaborate, as Caitriona showed you, with advanced liquid handling robotics and attention to how we prep all the samples. Even across dozens of sequencing runs, we can get this excellent data consistency, and we can also aggregate that across sites. Now, with our Green Genes 2 database, you can see it flipping back in the top left between WGS and 16S. So with Green Genes 2, we finally have a resource that allows you to integrate 16S and whole genome shotgun data. And we think this is very exciting from the perspective of being able to capture more information in a consistent way, including in settings where there's limitations on the cost of sample processing, so lower resolution techniques are required. But you can see this consistency over tens of thousands of samples. And to further increase representation, we partnered with Danone to launch THDMI, the Human Diets and Microbiome Initiative. And so here we were looking systematically across 5 countries, the US, the UK, Spain, Mexico, and Japan, to ask whether relationships between the microbiome and dietary response were consistent across these different countries. So again, this provided a significant international expansion using consistent methods, but Africa, unfortunately, was completely left out, as was South America. And in fact, by the time we'd got up to 25,000 samples in AGP, we had a pretty good representation from North America, good representation from Europe, and fairly good representation from Australia. But unfortunately, Africa was almost entirely left out. And I regret to say that we had so few samples from Africa that we pasted the example data over that continent on the map and South America. These are issues that we would really like to address going forward. Similarly, in the Earth Microbiome project, the initial plan for the project was that it was initiated by a large government agency, the US Department of Energy, in a meeting in a meeting initiated by Jack Gilbert, who was then at Argonne National Labs. But unfortunately, they didn't pick it up at full scale, and so we ended up doing it based on private foundations and in-kind donations from various companies, to whom we're very grateful. And so, as I mentioned, I co-founded this together with Jack Gilbert and Janet Jansen. Here we all are at the 2012 AAAS meeting in Vancouver, where we announced the project overall. And essentially what we did is we crowdsourced sets of samples from the scientific community that had already been collected and that answered an interesting spatial, temporal, or evolutionary question. And the scale of the project was remarkable. So back in 2007, Kathy Lozapone and I did a meta-analysis of all of the microbiome data represented as 16S data that was available in GenBank at the time. And so this covered 21,000 sequences from 202 samples, which was a total of all microbiome data collected by literally hundreds of labs. In the Earth Microbiome project, in the first main paper in 2017, we expanded this to 27,000 samples, so more samples than we had sequences just a decade before, and 2.2 billion sequences, providing a unique resource. And to do this, we had to develop a lot of the underlying techniques and protocols, many of which are still in widespread use today. So we were able to do a lot of really exciting stuff with the EMP dataset, including creating a new ontology where we could ask the microbes to tell us what factors were important to them using unsupervised clustering techniques rather than imposing on them our human notions of what was important. But regrettably, the geographic distribution was not nearly as even as we would have liked. And again, you can see that across sample types, Africa is underrepresented and indeed entirely absent from the saline communities that we looked at, which again is partly due to resource limitations on sample collection, partly due to information dissemination, and partly due to difficulties in international shipping. And the same was true for the EMP 500 paper. So this came out in Nature Microbiology 2 years ago, and in collaboration with Peter Dorrestein's lab, we were able to add a metabolomics layer to this analysis, as well as using shotgun metagenomics rather than 16S. And you can see just a couple of samples from Africa represented in the sample set. So we really need strategies to increase representation in Africa, and a really critical reason why we need to do this is progress in AI. So as I'm sure you're familiar with, AI is revolutionizing a lot of different fields, ranging from protein folding, where it's now able to produce structures almost comparable in quality to X-ray crystallography. That's true for weather prediction, where AI methods are able to do accurate forecasts at a tiny fraction of the computational cost of doing it with physics-based models. And at UCSD Health, we're even using it to rewrite communications from physicians to patients, where all parties report tremendous satisfaction with the system, which makes all those communications much friendlier, much more helpful and empathetic than they would otherwise have been. However, for AI, it's absolutely critical to maintain diverse training sets in order to avoid bias and also to avoid incorrect system behavior. And one example of this that made headlines a few years ago was the racist soap dispenser that was captured on video at a Marriott hotel, where essentially what the problem was that the manufacturers of the soap dispenser in Asia had only tested the infrared sensing mechanism using light-colored skin. And so darker skin tones absorb more light, fail to reflect enough back to the sensor to activate the soap dispenser, and therefore the automated system only works for some people and not for others. And this is kind of a frivolous example, but this kind of thing is increasingly pervasive in systems used in healthcare, where when the reference range is too restricted, the system does not work for all the people that it's intended to work with. And tying this to the vaginal microbiome, the dogma in the vaginal microbiome field has been that the healthy vaginal microbiome is dominated by lactobacillus. And that's true for 90% of Caucasian women in the United States and Europe, where the studies were done initially. But it's actually only true for about 2/3 of Hispanic and Black women in the United States. And in Africa, non-lactobacillus dominant communities are even more prevalent. So this is true even for healthy asymptomatic women. And what it means is that we need to substantially expand our range of what we consider to be a normal vaginal microbiome in order to set reference ranges for what's healthy versus what might need to be treatable. And the particular risk here is that is of defining people who are perfectly healthy as having a disease based on their microbiome being unexpected relative to too small a reference range. So there's a lot of excitement about personalized medicine these days as well, and we can indeed learn a lot from examining one single case. And if you saw my talk at ISME, you heard me talking about Larry Smarr. I'm going to talk about a different and I think even more exciting case here. This is work we did with Ruth Ley, who's now a director at the Max Planck Institute, where we were analyzing the development of the fecal microbiome of one child from birth up to 2.5 years, sampling the microbiome every week. And so this begins with the child's meconium, which, as you can see, is in the vaginal part of the plot, which is exactly what we would expect from his delivery mode, which has a huge impact on the baby's first microbiomes. And I'm going to start this going, and each frame in this animation is 1 week. And you can see sometimes there's a lot of change 1 week to the next. Other times there's just a little bit of change. And the question is, over those 2.5 years, how quickly does he approach the adult fecal state? How smooth is the progression? Is it rapid or slow? Does anything interesting happen along the way? And coming up in just a moment is something that we thought was very interesting and indeed quite shocking. So what happens is he gets antibiotics for an ear infection. You can see this amazing regression of his microbiome followed by a rapid recovery. And that went by pretty fast, so I'm just going to rewind it for you and play it again. What you can see is that on administration of oral amoxicillin, there's this tremendous regression of the microbiome, undoing months of normal development, but then he bounces right back to where he was in just a couple of weeks. And by the time we get up to 2.5 years, he's in the healthy adult region of the plot, as defined by the Healthy Adults in the Human Microbiome Project. So this child was resilient to antibiotics, but we don't see that in every child. And trying to understand who's at risk and who's not of having their microbiome permanently altered by antibiotics, as well as restoration techniques afterwards, as a continuing focus of work we're doing with Maria Gloria Dominguez-Bello, who we did the initial work on vaginal versus C-section deliveries and their alteration of the microbiome with. So that's just one kid though, and generalizing to larger populations can be hard when you're looking at these complex trajectories. But when you view the data the right way, sometimes you can see very clear patterns. And in work we did with Jeff Gordon's lab back in 2012, led by one of his very talented grad students, Tanya Yatsenenko, an African population in red, a South American population in green, and the US population in blue. the convergence to the adult state is exactly the same for the three populations, and most of that convergence happens in the first 2.5 to 3 years of life. So what we're looking at on the x-axis is age, and on the y-axis, the average distance between each point and the adults in the same population. So that looks like a consistent trend, but then where they end up is completely different. So these are principal coordinates plots of the same data, where you can see in the left-hand set that age is the main driving factor, but in the right-hand set, what you can see is that the populations end up completely different from each other, with the US being profoundly different from the other two populations. And when we think about large-scale efforts like the Human Microbiome Project, it only looks at healthy adults. And as soon as you look outside North America, and as soon as you look at children, you see these completely different configurations of the healthy microbiome that it's critical to take into account if you're trying to tell the difference between health and disease. And this is true for the skin microbiome as well. So this is additional work we did with Gloria, comparing various non-Western populations to the US. And as you can see, the non-Western populations all cluster together and are completely different. So we're increasingly recognizing this is true across body sites. Coming back to the stool, in work we did with Justin Sonnenberg and a number of others on the Hadza hunter-gatherers in Tanzania, what we see is in people living this non-industrialized lifestyle, a tremendous amount of seasonal cycling that we see in very attenuated form in the American Gut Project in the North American population. And what was amazing when we looked at the principal coordinates results across populations is that along principal axis 1, the different populations automatically sort out by the level of industrialization. And this industrialization process is coupled to the loss of a whole lot of major microbial groups that we've co-evolved with, as well as the gain of a couple of essentially weedy taxa that lead to a much lower diversity community. Now, I regret to say that although this initial paper did include several authors from Tanzania, in subsequent work that I wasn't involved in, done by Justin's lab, all of the African authors were dropped on continued analysis of these samples. And I think it's really important to maintain communication when you're doing subsequent work on sample sets with the people who collected the samples and designed the research, rather than considering their work done once you have the samples in your lab in the US or some other country. So what's needed to train equitable microbiome AI models is indigenous data, and the studies have to be designed and the data have to be collected and analyzed with indigenous input. And it's especially important to get input from the communities that you want to study on the problems that are most relevant to them that you can potentially solve with your research, and approach this in a collaborative way rather than in an extractive way, where all you want is the samples, and then you're going to carry out your preconceived research program on those samples. And there are some efforts to address this kind of thing in progress already. So the Microbiota Vault, led by Maria Gloria Dominguez-Bello, is a global nonprofit initiative to conserve long-term health for humanity by preserving microbial biodiversity. So, so essentially the idea behind this is to back up local microbiome collections in a permanent archive. And we outlined the need for this in Science back in 2018, and progress towards it has been substantial. And the idea is that the provider of each sample retains ownership of the sample and any IP and control over what research is done with it. And the Microbiome Conservancy, another effort led by Mathilde Poirier and Mathieu Grossan at the University of Kiel, is similarly doing this sort of thing specifically for human microbiome specimens, including a culturing initiative. And as you can see, they've managed to get fairly good representation across Africa, although there's still a long way to go. So there's techniques in AI like transfer learning and upscaling, where you can apply information from massive international databases to much smaller African data sets, although this is very much work in progress, and the requirement for that training data is still critical, because you can extrapolate from a relatively small amount of training data that you've collected in a population relevant to what you study, but you can't extrapolate from zero. I'll just close with this quote from William Gibson, the science fiction author, he's quoted as saying, "The future is already here. It's just not very evenly distributed." And we're already living in a future where microbiome research is delivering amazing benefits to patients in terms of stratified drug prescriptions, in terms of therapeutics that are approved in the United States and in Europe, for example. But those benefits are not yet being realized in Africa to anything like the same extent. And it's critical that we build up in-country capacity in countries across Africa for sample collection, sample processing, data analysis, research design, in order to make sure that these benefits are more evenly distributed in the future. And with that, thanks again for a great symposium and for inviting me to it. Again, sorry I can only join virtually, but certainly looking forward to my next visit to Cape Town. And I'd be delighted to answer questions with the rest of the panel. Thank you.
Thank you very much, Rob, for the wonderful presentation. Could you please stay on the call for the questions from the audience? And unfortunately to everyone else, Rob's talk was the last talk. It would have been nice to have another 10 more presentations. Joanne, next time, please, let's have a lot more presentations here. And at this time, I'm going to welcome all the presenters that have presented today to the front. So we are going to have questions from the audience. Yeah. There's a question from Joanne.
So my first question goes to Moses. And I really appreciate that you took up this opportunity to talk to us a little bit about your thoughts around the impact of AI. On previous meetings, I've actually heard you talking about the metabolomics, the proteomics, and the microbiome data you're going to be getting from your pregnancy study. Can you talk us through a little bit about what the objectives are about synthesizing that and maybe the impact, why you're doing the study?
Thank you very much, Joanne. And so I took quite a generic look at the AI approach in African Indigenous data. But what I didn't mention is about my work that is focused on vaginal microbiome and metabolomics. So I'm currently in Kenya as part of my Callisto Juma Fellowship. I've been able to recruit about 1,502 women in pregnancy from first trimester the way to the third trimester with pregnancy outcomes. And the aim of this is to look at the change in the vaginal microbiome with the various pregnancy outcomes. So what we are doing is we have women with preterm birth, and in our study so far, we've had about 90 women who've had preterm birth as an adverse outcome. We've had about 60 women with stillbirth. Then we've had over 120 women miscarriages and others with the intrauterine growth restriction. And then the rest of the women with who either dropped out of the study or had a full-term pregnancy. So we'll be looking at what's the relationship between these pregnancy outcomes and the vaginal microbiome in the first place. Then we'll also be looking at what are the metabolomic profiles of these women and how they relate to the pregnancy outcomes, and then how we can use this data, metabolomic data, microbiome sequencing data, clinical data, and put them on a machine learning platform to be able to predict these pregnancies that are at risk of adverse pregnancy outcomes, and furthermore, also to see if we can use some of this metabolomic data to plan a mini panel that can be used as a point of care screening tool or diagnostic tool for pregnancies that are at risk of adverse pregnancy outcome. So in very brief summary, that's what we're doing in our country, and that's how we'll be applying the machine learning platforms and metabolomic data to improve pregnancy outcomes.
Thank you very much. There's another question.
Hi, Caroline Mitchell from Mass General in Boston. And I think this is a question for all of you. So much of the dogma around the vaginal microbiome is things we've said for decades that I'm not sure are backed up by mechanistic data. And so there's work to do to incorporate more data from Africa. There's work to do to incorporate sort of mechanistic work from bioreactors, like what you're doing, what Andriati's doing. And what we do know about the microbiome. And I guess I'm wondering, how can we is it possible to use machine learning and AI to put all of that together? Data from mechanistic studies, data of things we know, data of things we think we know, to expose more of the gaps. What are the things? Where are the places? Just like looking at the sort of maps, the spaces, and the computer saying, here's where you need to look more. Can we do that for the field of the vaginal microbiome, and specifically the African vaginal microbiome, to say, what are we missing? Where are the spaces where we really need a lot more data? Is that possible?
I don't know who's going to pick up that question.
I'll just comment quickly, and
Sure.
then I'll let the AI experts finish off. But some gaps that I've seen from just doing some reading is basically, like, I'd love to see more paired sample collection between fecal samples and the vaginal samples, because of the really fascinating estrogen gut microbiome axis. And I'd love to just see more correlations there between the changes in the vaginal microbiome and the gut microbiome, and how maybe then that could inform dietary interventions and who knows what else. But it will definitely help on the AI side as well.
I'll take a stab as well. I mean, this is probably a question more for Rob than me, but one of the things that I've become obsessed with is changes in microbiome over time, well, changes in metabolome over time, changes in just periodicity. And one of the things that, if you look at clinical analysis, there's an awful lot of clinical measures that you have to do at specific times of the day because people have different rhythms and things like that. So I wonder in a lot of cases whether you're getting big, and as Rob pointed out, you're getting these big changes in the microbiome in seasonals and stuff. Does that happen in all sorts of other conditions as well?
Sure. So
Rob,
the short
would you like to
answer,
comment on that question?
yeah, absolutely. The short answer is yes. So one of my postdocs, Celeste Alaband, has done some really amazing work looking at daily fluctuations in the gut microbiome, and in both humans and in mice, the time of day that you collect the sample has a huge effect both on the microbiome itself and whether you see differences between groups in terms of their microbiome. Where if you look at if you look at midnight, maybe you'll see a large difference, but if you look at midday, maybe you'll see no difference at all, or vice versa. So understanding those rhythms is going to be really important. That's one factor that limits our ability to interpret current data with AI. Because time of day isn't even recorded in most microbiome studies yet, unfortunately. The other big issue is representation. So as far as I'm aware, the data sets that have been used for training on more mechanistic and especially physics-based models are much larger than the data sets that we have access to, especially for vaginal microbiomes in Africa. And also the data sets tend to be much more AI ready in terms of being collected with consistent methods like you saw from Caitriona on the lab side, and also being annotated with consistent metadata. So even with advances in natural language processing, I think it would be a considerable challenge to use AI to take what we've got now and turn it into a model, even for the purposes of gap highlighting. I think there's just so much gap at the moment and so little data that it's perhaps not ready to address with AI yet. Sean, what do you think? I know you've thought about these issues with respect to multimodal data integration, especially.
Yeah, I don't have a ton to say on the vaginal microbiome, honestly, because I don't think about it that much. But my first guess as to where would you make measurements to improve our current knowledge set, I've become more biased that much of what the microbes are doing is metabolic in nature. So pairing metabolomic measurements with microbiome composition measurements and more systematic measurements across the body is probably useful. And also immune interactions. So looking at interactions between peptides and proteins derived from microbes and host receptors. I think that's another area to zoom in on. And then from a timing perspective, I'll add my little 2 cents there, where we also did some work when I was in grad school to look at mice and diurnal patterns in the gut, which you definitely see, but people poop about once a day. So it's actually hard to do a time series of humans in the same way as you can with mice. And what we see in humans is that fluctuations tend to be regressing to the mean when it comes to the average abundances of the major taxa over time on like weeks to month timescales in humans. So I would guess that your microbiome today is going to be pretty similar to your microbiome 2 or 3 weeks from now. You can push it in a direction, but it takes months, many weeks to months for it to move essentially. And that's the gut.
Another question.
I have another question.
Sure, go ahead.
So this one is to Rob, but if others have thoughts, I really appreciate the effort you put into highlighting, the samples in global databases, and particularly, these major initiatives that are global and highlighting that South America and Africa were not part of those discussions. From your view, what would make it easier for partners in the global north to include African collaborations? What is how can we do this better so that we are ready to contribute?
Right. Well, we did try quite hard to get representation from African and South American groups especially in the EMP. I think the challenges were, so our policy with the EMP was that we would do the sequencing, data release and data analysis for free, but the submitter was responsible for collecting the samples and getting the samples to us with metadata formatted in a way that allowed integration with the rest of the project. So the main barriers there were either people had really cool sample sets that they aspired to collect, but then the funding fell through, or there were logistical or transport issues that prevented the samples from actually being collected, or the other big barrier was shipping costs, where in that project we were not funded to support the shipping costs, but in other projects where the funding agency explicitly does support the shipping costs, that has been a lot easier to get samples from. So, for example, in our Gates-funded project in Bangladesh, looking at malnutrition, for example, the Gates Foundation just pays for all the shipping. So the high cost of that and inconvenience of that is mitigated by funding. So the reason why we have that policy is that it's really difficult to transfer funds between institutions, and funding agencies that are able to facilitate relatively small but still essential amounts of money to go to international partners for those kinds of logistical details. That's something that in practical terms makes a huge difference. I don't think there's any trouble with workforce training or capacity, like the metadata that we got on samples from Africa was always well executed and done to a high standard. And now that video conferencing is so prevalent, the ability to build up that capacity and get it exactly right is much easier than it has been in the past. So I think I think the main issue is funding, and also allocation of that funding to the African or South American partners in the ways that are essential for the process to work end to end.
Thank you very much. So we are out of time. Can I ask one more question? Convey one more question from someone who's attending the symposium online. So this is a question for Sean. The question is, what was the reason for not including Africans in the Diet and Microbiome Initiative? And is it too late to include African scientists as part of this initiative?
I actually think that's a question for Rob.
Yeah, I think that's a question for me. So the reason why is that the countries were chosen by Danone based on their market size, and we pushed quite hard for a broader representation in the initial 5. They're currently evaluating whether to extend the project further. Each country that the project deploys in does have local partners. So if they are willing, or if some other company is willing to extend the project to international sites in Africa, then yes, absolutely, African scientists will be involved in those components. And the data set will be open, so like fully open. So for reanalysis, it will certainly be possible to get involved in that if you're interested.
Thank you very much. Thank you very much, everyone. Thank you, Rob. Thank you, Sean. Thank you, Moses, Karl, and Caitriona. Yeah, now I yield my time to Joanne. Unfortunately, I have to go back to my friend, the cursor that is blinking on my computer.
Thanks again.
Thanks.
So that was a phenomenal session to end with. And I think we're all very inspired by the possibilities that AI and machine learning can bring. Is this on? No. So with that, I would like to start the closing, and thanks. And I would like to invite my co-chairs up to the front to put them on the spot. I wasn't very hospitable when I started this meeting, but I could not have done this alone. And I've got 3 very powerful co-chairs in Lenine Liebenberg, Anna Happel, and Heather Jaspin. You are seated. Just for those online, but also in the room that haven't heard enough about vaginal microbiomes, I wanted to put up this advert for a seminar that Lenine is leading tomorrow at SERI, Stellenbosch University, in which Caroline Mitchell, Doug Kwan, and Jacques Ravel will be talking there as well. And I look forward to attending that meeting as well. So the closing session, from my perspective, I wanted to circle back to what I talked about at the beginning of this meeting, for this vision that we put to this audience for an interconnected global database abundant with African microbiomes, shaping a better future for human health on the continent and globally. And to do this, we need massive acceleration of the way we work, and we need good enabling partners. So I outlined some of the challenges, and we heard yesterday from Tulani and Voke about some of the gaps in the current ecosystem. And for the purposes of this meeting, I don't want to spend too much time on the challenges, but I would rather focus more on the opportunities. I'm just waiting for the slides to proceed. So the way I see the path from this week, the discussions over the past 2 days for accelerating African microbiome research, centering on the pillars we identified yesterday about local scientific leadership and African governments. So some of the points we raised yesterday in my opening was we need this local scientific leadership to drive robust research agendas, to coordinate intra-continental collaboration and network. And I think that's, to me, one of the pieces that we've really sorely missed the opportunity for in the past. And I think we're addressing it slowly, but we need to accelerate that as well. Establish global strategic partnerships to enable this relationship building between the South and the North, and prioritize infrastructure development, including focusing on next-gen sequencing. And then central to everything, all 4 of us co-chairs are passionate about is training, nurturing the next generation of African scientists. And then from our governments, we need them to commit to further strategic investments in microbiome research to build on the amazing support they already give us, and promote policies that accelerate ecosystem building. And I think, to me, some of the elements and advice from Rob Knight in his response to my question highlight some of those key pieces we can start having conversations with our governments about to enable Africa to be at the table and contribute. So I want us to think about actionable next steps to accelerate microbiome research on the continent and what that looks like, and then actionable steps to establish South African Microbiome Research Hub. So we asked each of the session chairs to just pick the strongest take-home message from their session, and I'm going to need the roving microphone, please, so that I can go through each person respectively. And if I was to take one message home from my session as being the first session chair, is that the messaging came through clearly from all three of the speakers, was the importance of focusing on women's reproductive health and how it's completely appropriate and long overdue. And really, we need to amplify research around that. I'm going to ask Lenine to speak next from your session. What was your biggest take home?
Okay, besides the importance of getting around a lot, and I will say that again and again and again, how important it is to diversify your perspectives, your experiences, interdisciplinary research, just adds into how we are able to problem solve better in a more inclusive nature. I started off with besides, so I've got an additional bit, right? I want to emphasize the importance of the kaleidoscope analogy that I mentioned, those little differences in colors and patterns, and getting researchers, governments, citizens, funders, everybody involved to prioritize the microbiome research agenda, and particularly for women's health. I think those were the key points for me.
And then Anna.
Sure. So the focus of session I chaired was diversity, and I think we all acknowledge that there's a lot of diversity in terms of languages, cultures, genetic diversity, microbial diversity. And I think the key point that I took from the session was that in order to unravel the microbial diversity and its meaning for human health, we need both scientific knowledge, but also traditional ecological approaches. And we need to work together and collaborate between scientists, policymakers, and communities, and really adhere and acknowledge that we do this in an ethical and equitable way.
Perfect, thank you. Nikki, Tiffin.
Thanks. Yeah. So I think from our session, what was very clearly articulated is that first of all, that microbiome research with African populations is currently very underrepresented in global microbiome research, but that it has this capacity to really generate huge benefits. And that if we can ensure equitable international research relationships, this can support African researchers to truly lead and run their own research in their own countries with the understanding of the local context, local benefits. And then, just to say, equitable practices can ensure that African populations can benefit from this kind of work alongside the global populations. And that African researchers can increase their research capacity and sustainability of their research on the continent. But the messaging was very clear that nobody's saying they don't want to collaborate, and nobody's saying that they don't want to benefit global populations. They want to have global interactions, global collaborations, global benefits in which Africans are included as equal partners and can participate fully and benefit fully. Thanks.
Perfect. Heather Jaspin.
Thanks. I think what I'm thinking has been already said a little, but from my session, I think my wish would be that we had a Sala Sisterhood project in every country in Africa.
But the South African one is called the Mary Project.
The Mary Project is...
We're going to get it funded.
But that's one of many, many countries that needs one that has engagement with the population at the very beginning, and that not only has a description of the vaginal microbiomes, but also the metabolomes and what's driving the mechanisms, such that we can develop socially appropriate diagnostic, prevention, and treatment modalities from this information.
Thanks, Heather. And then Sarah. Sorry, Sarah.
Thanks, Joanne. I think from my session, my takeaway was that the future of vaginal microbiome manipulation is bright, be it in transferring single strains of lactobacilli or multiple strains, or the whole community, the lactobacilli and their friends, or even using phage therapies to, decrease the resistant bacteria. So there is potential for synergism in the sense that we can use multiple strategies. For example, what Sinai was talking about, using the phage therapies to clear the resistant Gardnerella and then bringing in either the transplantation or the probiotics. There's also potential for synergism in terms of working together both within the continent and across the different continents. I think in terms of accelerating these products these innovations into products that women can use and can access in the market, we have to think about contextualizing that research for Africa, looking at the different needs, the different contexts, and ensuring that those therapies can work for African microbiomes. Thank you.
Thanks, Sarah. And then last but not least, my favorite machine learning expert, Sinkala.
Thank you very much. Most of the things that I wanted to say have been said, but I will say something. What I've seen and what I've taken from the entire symposium is that there's a lot of research that is taking place, starting from community engagement to laboratory experiments that are being conducted to modeling related to microbiomes. And what I've seen there is that there is room for collaboration. There are a lot of people who have got skills in areas where other people don't have skills. And maybe it would be a great idea for those people to come together and collaborate where one has a weakness, to collaborate with other people who have some strengths in order to compensate one for each other. Thank you.
That's perfectly put, because that is our hub. So, yes. And then I wanted to give Sonia if she's ready for me. And okay. Sonia's going to talk us through her graphic harvest of the meeting, which I hope will echo some of these points that the chairs raised.
Thanks. So I'm not going to take you through everything because there's so much here, but I'll give like a high level so you get a sense of the landscape of it. So it flows from left to right, and everywhere we see one of those big circles on a heading is kind of like the big theme under which usually around 3 or 4 kind of talks were clustered. So the first one here was just about restoring balance. And so you have kind of drawn the world and shown kind of that missing puzzle piece, which is the African microbiomes. And we have a distorted picture because it's missing. And also having the greatest sort of diversity of thought and culture and actual microbiome data will allow us, instead of doing this one-size-fits-all approach, so the umbrella approach, to much more precision approaches. It's like the kaleidoscope means that we can actually target our interventions much more carefully. Another piece about restoring the balance is that health research is very much skewed. We have underrepresentation around research on women. And so now all the medical words are leaving me. But there's a disparity in the research. There's a lack of effective treatment and lack of personalized healthcare. And then there were statistics given. So thousands of women die daily from preventative reproductive diseases. And then we see a huge impact specifically in sub-Saharan Africa, where the young women are disproportionately at risk for HIV. So it's a real target community to focus on. And we were exploring, well, what can we do? And sort of from some of the Q&A at the end, really the pieces that came out were sort of education, empowerment, and giving women choice, so acceptable and desirable prevention options. So there was that whole train of HIV prevention options we spoke about. We also spoke about the launch of MAL, the Microbial Interactions Lab, so I've got the little logo there. Then the next piece was around collaboration and partnerships. So that's kind of all sitting down here. So we looked at, as was just mentioned, we need more of this kind of ecosystems of research, intercontinental and intracontinental. So how can we create enabling environments so that that's kind of metaphor of building your stable and then breeding your own thoroughbreds and then attracting all the brains from the rest of the world, repatriating them and bringing them here. To do that, we must create great facilities people can do high-level science at. We need the right kind of infrastructure so we can manage big grants. And also it's about sharing the infrastructure. So it's not about everyone having their own awesome lab, but it's about sharing so we can actually get the outputs we need. Then it's about holistic collaborations instead of just sharing information. How do you do it in a deep way? And that was kind of what Suri shared. So how can we share knowledge, programs, protocols, analysis, and then just build the capacity? So if we have researchers kind of all over the world, if they see unusual outbreaks or maybe they're collecting interesting data, how do we support them by sharing protocols, sharing reagents, really going much deeper than just knowledge is what we always end up doing when we talk about collaboration. And then governments, how do we get them involved? How do we get them interested? So how can we speak to their policy interests and get them to invest in our work? So that was that piece. Then here we got into the African diversity. Here, Africa is not a single place. There's so much diversity throughout, and we need much more nuanced approaches. Here we've got robust ecosystems, and so here we spoke a lot about the ethics of the research and how can we create more equitable partnerships so it's not so skewed to the global north. And I think a big piece that came out in there, just trying to see where I wrote it, was just the strength in numbers. The better we collaborate, the stronger we are, and then we can actually have some leverage in these negotiations and at the research table. And so there are lots of just sort of little pieces of what that more collaborative, equitable research could look like. And you can really set that up by design and how we agree on things and have contracts and agreements around it. And then on this side is everything that happened today. So I won't go into it as much. Here we've got sort of studying the vaginal microbiome. Here we had the citizen science example, how important it is to have really clear narratives and really, really good communication, and not just have your medical scientists, but also social scientists and communication experts in terms of that messaging. Also how to contextualize it, so make sure you're working with people who communities trust, even with peers from the community. Here we've got I don't remember this, but I'll come to it later. Here's all the novel treatments. I've drawn the kind of holy grail with the Lactobacillus crispatus as the dominant. And then here I've written, as far as we know, that's what a healthy biome looks like. But in women from the US and Europe, you know, what else could it be? But that's kind of what we know. So here we've got the vaginal microbiome transplant. So here I've drawn the Lactobacillus crispatus looking very friendly and all of his friends, and they're ready to go over here. And then here we've got the Vibrant trial. So here we've got the different strains of crispatus. So here we've got a whole bunch of the friendly guys in a pill, 6 of them, and another one, 15 of them. And they want to go and colonize. And then here we've got the PM477 and the phage therapy. So here we've kind of drawn the Gardnerella and his biofilms and all the other nasties he's harboring in there, and so trying to show that this work is trying to create an inhibitory effect so that antibiotics can work better, or our probiotics can work better. And then the last one is just on the AI, machine learning and informatics. A lot of that was beyond me. This last session was very detailed. I did my best. But here just really, I can hear it just dramatically speeds up our processing power and how quickly we can get information out by vast quantities. And then a big theme is, so there's all this great data, but if the data sets are biased, we're going to get the wrong kind of information, bad diagnostics, bad predictions. And so we need to make it much more inclusive. So we need to make sure that our data sets are designed, collected, analyzed with a local input. And for that, we need to invest in the Africanization of AI. And then here we just had some more specific lab sort of methods for collecting samples, and also crowdsourcing. So how do we get it out from the clinical setting, sort of in a general, sort of more diverse setting for greater diversity of our samples? And I don't know, I've had a blank on that one. Oh yeah, it's really just about the dysbiosis. We often just base diagnosis on symptoms rather than lab testing, because it's often very expensive, all the testing. And we also easily just over-treat with antibiotics and antifungals, which creates more dysbiosis and problems. And so there's a huge need for more good diagnostics. And then that comes into all of this new terrain, which is the novel treatments. Okay.
So Sonia's present to us and to each of you is a high quality capture of that information that you will have available and can love. I've had Sonia do another project with us, and it's one of my favorite things to look at all the time. So, and that will be framed somewhere in the microbiome lab, and we're going to be very proud of it. Thank you. So in ending off, I just wanted to take us back to the purpose of us coming here. Africa is well positioned to lead the charge in reproductive health, research and innovation, and to innovate locally driven solutions that address global health challenges. And I think we've had a really rich dialogue with collaborators locally, across Africa and globally, Europe and the US, to help us get there. So we really appreciate the 2 days that you've spent helping us map what this could look like. And my final vote of thanks, if I am allowed,
is to thank the Institute of Infectious Diseases and Molecular Medicine for believing in this initiative, for creating the space and the resources to start this process. It's with huge gratitude, and I think we're going to do them very proud, and we're going to shine. And Janine, Anna, Heather, myself are going to have a real indaba soon, mapping out how we're going to take over South Africa's vaginal microbiome center of excellence. So watch this space. Thank you very much.